Hello, everybody. Firstly, I would like to thank the organisers for inviting me to come and speak at this meeting. What I want to do today is I want to talk to you about the research that I've been doing over the past 10 years, whereby I've been looking at the biomarkers of aging in bats to try and uncover what bats have evolved to allow them to slow down the aging process and really achieve these extended health spans. Hopefully, I'll be able to take some questions at the end of this recorded session if the internet from my field station in rural France is up to scratch. But why should we study aging? Well, really, a grand challenge society faces today is healthy aging. We urgently need to develop a better understanding of the aging process with a view to finding ways to alleviate it so that we can extend our health span. But why do we need to do this? People everywhere are living longer. It has been estimated that a baby born today will be able to live until she is at least 150 years of age. Most likely she's alive in Japan. But we've got a problem. And the problem is, despite our increase in expected lifespans, we have not increased our health spans. There is going to be an increase of approximately 340% of people over the age of 80 in the next 30 years, and a doubling of people over the age of 60. But our probability of acquiring disease of the old age by the time we're 60 has still stayed the same. These are diseases such as Alzheimer's, dementia, cancer. So if we want to be able to match our health span to our expected lifespan, we have to find ways to understand aging. We have to find ways to understand how we can age healthier for longer. Because if we don't match our health span to our new extended lifespan, our future societies are going to be full of the incapacitated elderly. But we've learned an awful lot from studying the aging process in model organisms such as mice and flies and nematodes. But potentially the problem that we face right now and why we don't understand the aging process and why we don't yet know how we can age more healthily is that we've been looking in the wrong place. These model systems have given us an excellent insight into trying to untangle the complexity of the aging process. But let's think about it. These species were chosen because they can die well rather than live long. Wouldn't it be much better if we could study the aging process in species that have naturally evolved longer health spans, such as the bats? So it was this image that was published by Steve Asset in 2010 that really stimulated and drove my passion for studying aging in bats. Typically in nature, there's a pattern or nearly a law. Small things live fast and die young. Big things live slow and live long. Think of a mouse or a shrew living six weeks, 6 months, maybe 4 years, or a bowhead whale living over 200 years. So you can predict how long species live for given their body size, for the most part. But bats buck this trend. So this is a chart looking at the expected longevity given body size to actual longevity for over 600 species. And what you find is that there are 19 species of mammal that live longer than humans, given their body size, and 18 of these are bats. And these are wild bats that we've studied with microcapture studies. One's a naked mole rat. Now, what this suggests is that bats have evolved mechanisms to allow them somehow slow down the aging process. And indeed, the bat that holds the record, the longevity record, is Myotis brandtii. So there was an individual that was caught in Siberia, a male, in the wild, had a tag put on them, was released, and was caught 41 to 43 years later with no signs of aging. But what is extraordinary is not the 40 plus years of age, but the fact that this species, they weigh less than a third of a lab mouse, but they're living over 10 times longer than a lab mouse possibly could. Lab mice can never live longer than 4 years unless you do some major interventions. So this suggests that bats have evolved mechanisms to allow them overcome the expected aging process. So therefore, studying bats can allow us uncover the mechanisms that mammals have evolved to have extended health spans. So with this in mind, for the past decade, I've been developing bats as an alternative model system to uncover the molecular pathways that underlie extraordinary aging in mammals. And I've been studying and developing and uncovering the biomarkers of aging in long-lived bat species to ascertain what bats have evolved to allow them to slow down the aging process with the view to translating these understandings into extending human health span. But how do you go about doing this? How do you go about looking at aging in a non-model organism? Now, firstly, you need to have exquisite reference quality genomes. And when we started this project 10 years ago, there really were no decent reference quality genomes for bats. So to overcome this, myself and Sonia Vernes founded Bat1K. And Bat1K is a genome sequencing consortium, bringing people from all around the entire world with a view to sequencing the genome of every single living bat species to chromosome level error-free reference assemblies, so that we can uncover the unique adaptation that bats have evolved, so that we can look at the biomarkers of aging, design assays, and study bats in an aging context, for example. So we published our flagship paper in July last year, whereby we sequenced the genome to reference quality of 6 different bat species. We developed the methods to sequence, to assemble, to annotate, to analyze these genomes. And we included genera of the longest lived and the shorter lived bats. So we have access now to good quality genomes that allow us to look at biomarkers of aging in bats. But to really answer the question of how do bats defy the aging process, we need to look at what happens in bats as they age. Now, it's extremely difficult to keep the longest lived bats in captivity. These are your temperate insect-eating bats because they don't do well. They don't have babies. And typically, aging studies would study individuals as they age from the day they're born to the day they die. So to try and study bats in the aging process, about 12 years ago, I wrote an ERC grant working with this fantastic group of conservation biologists in France called Britannie Vivante, with some amazing collaborators. And we identified a population of the longest lived bats. These are myotis bats. Their maximum lifespan has been estimated at approximately 37 years of age. And this population has been studied in France for the past 20 years. Individual bats have been captured as babies. Transponders have been put inside them from the year that they were born. And what we do is we go back to the maternity roosts that the females return to year after year after year. So we know the age of the bats because an individual that was caught as a baby in 2010, when we catch them again this year, we know that the individual is 11 years of age. What we do is we designed assays whereby we can non-lethally sample the same individual year after year after year. We take less than 100 microliters of whole blood. We take a 3mm wing biopsy punch. We take anal swabs to look at the microbiome, sometimes mouth swabs. We measure the weight of the individual, the quality of the individual. We catch the entire colony every single year in France. This is why I'm in France right now. Feed and release the individuals. And what this allows us to do is this allows us to track and look at biomarkers of aging in one of the longest lived wild bats to uncover what happens in bats as they age. And to date, we've been looking at the changes in these biomarkers between young, middle-aged, and older bats. And so we have the genomes. We now have a fantastic population that we can study every single year. But we had to develop the different biological assays to be able to look at biomarkers of aging in bats. We firstly looked at telomeres. Remember, these are the protective caps on the end of the chromosomes, TTA, GGG, that shorten every time a cell replicates. We want to look at the mitochondria. Bat fly, they have the highest metabolic rates of all mammals, who consume huge amounts of oxygen with this very, very metabolically costly activity. We wanted to ask the question, do bats experience the same level of oxidative damage in their mitochondria as would be expected given their high metabolic rate, or they evolve mechanisms to maybe somehow stop that? We wanted to sequence their entire blood transcriptome. Blood is a great proxy for overall processes that are occurring. You can find 6 to 80 of all protein coding transcripts in whole blood. And we wanted to ascertain, do bats show the same level of dysregulation as they age as we do? Or do they have some other ways of maybe not experiencing this? I wanted to look at their microbiome, look at the overall change in their gut microbiome to see, do bats show some different signature? We want to look at autophagy. As we age, our ability to remove cellular damage decreases. Have bats evolved a different mechanism? And again, we want to look at their innate immunity. Do bats have some way of modulating inflammation that would be expected given aging? So I'm going to give you an example of what we did with our telomeres, and then talk you through some of our other results. But let's remind ourselves what telomeres are. The cell's machinery cannot replicate to the end of the chromosome. It can't add on to the 5' end. Therefore, every time a cell divides, your telomeres get shorter and shorter. Eventually, when they become so short, that signals that this is an old cell and the cell should self-destruct. This drives the aging process because sometimes those cells do sit there, these senescent cells that can't replicate. They emit SASP, they cause other cells to age, and this potentially can drive the aging process. So what we wanted to do is we wanted to modify this qPCR assay designed by Cawthorne for humans to work on bats. So we could look at the relative length of telomeres in bats as they age and compare the relative length of telomeres in young, middle-aged, and older bats. And this was work that was designed by Nicole Foley, whereby you see here's our qPCR assay. If you have a signal early on in our cycle, it's a long telomere relative to a single copy gene. If it's later in the cycle, it's a short telomere relative to our single copy gene. So we developed this assay that we could non-lethally sample bats. We could take wing punches every single year. And this was different because typically telomere assays used to require a huge amount of DNA. And this didn't work if we wanted to have non-lethal sampling. So we looked at our long-lived myotis bats, but we also worked with the bat community to look at other long-lived bats throughout the world. And again, these were excellent marker capture projects. So we looked at long-lived myotis in Brittany. We had long-lived myotis specksteinae in Germany. Rhinolaphus firmoquinum, one of the longest running marker capture projects, has been going on over 60 years due to the dedication of Roger Ransom. We also were able to look at telomere length and how it changed in Menioptera shrubbericea. So to try and do this right, you need longitudinal marker capture field studies and new molecular methods. So what did we find? What we found was extraordinary, is that really telomeres do not shorten with age in the longest lived bat genera, which are the myotis. They do shorten in Rhinolaphus and Meniopteris, but not in the longest lived bat genera. So we were very interested in, well, how come? How is this possible? We had looked for evidence of telomerase being expressed, and it wasn't. So instead we went and we looked at our genomes and did a comparative genomic analysis of 225 telomere maintenance genes. And we looked in myotis bats, was there any evidence of selection occurring? In reality, there was no evidence of positive selection after FDR correction, but there was evidence of divergent selection happening at ATM and SETX along our myotis branch. We also compared the overall transcriptome profile of bats to other mammals that were available. And what we found is that the myotis bats were enriched in DNA repair mechanism, and potentially bats were using alternative telomere length pathways to maintain their telomeres. So really we found there was no relationship between telomere length and age in myotis. Telomerase was not expressed in myotis blood or wing transcriptomes. We uncovered by doing phylogenomic analysis that bats seem to show this enhanced DNA repair, selection, and expression. And potentially bats are using alternative telomere length mechanisms to maintain their telomeres. We next looked at and sequenced the entire population of mitochondria to try and find, when we looked at the level of heteroplasmy you find in mitochondria, did we find any evidence that in young, middle, and older age bats that they were experiencing the level of oxidative stress damage you would have expected from their heightened metabolism? And what we found that it was not the case. Bats did not show this high level of oxidative damage as would be expected, given the fact that they have an increase in oxygen consumption, sometimes 130-fold higher than other mammals of the same size. So this suggests that they're able to repair or remove damage to their mitochondria. When we looked at their microbiome and sequenced the entire gut microbiome, we found that in bats, in young, middle-aged, and older bats, they did not show a change. So they're able to maintain homeostasis. We also sequenced the entire bat blood transcriptome because what we wanted to look at was the bat transcriptomes, find any age-related transcriptional changes. We also wanted to look at these small microRNAs that maybe were regulating these transcriptional changes to uncover what adaptations of extended longevity do we find in bats. Again, we looked at young, middle-aged, and older bats. What we found was extraordinary. We were able to compare the blood transcriptome from bats to humans, mice, wolves, because these were actually available at the time. We found that as bats age, you can see here, this is increase in expression of these pathways as bats age. In red, blue is a decrease. But bats increase the maintenance of their DNA as they age, and this is different from other taxa. We also found that really, doing our analysis, taking blood, sequencing the entire transcriptome, looking at young, middle-aged, and older wild bats, we found that bats show similar expression with age of genes known to extend lifespan in model organisms. For example, here's P10. If you're knocking a second copy of P10 into a lab mouse, they will live longer. But bats have naturally evolved the expression profile that we can see if we extend lifespan in model organisms. This means that our methods worked, thank goodness. But so what? We already know this. We've identified a whole series of potential new candidates that may be leading this. The other things what we uncovered is that potentially these longevity signatures are regulated by microRNAs, these small regulatory microRNAs that maybe are driving this. So we were looking at myotis, which were the longest lived bat genera, but we were then able to compare the blood transcriptome with molossus, which is the shortest lived bat to date. It has a LQ of 1, which means it lives as long as expected given its body size. And when we compared 8 adults' blood transcriptomes, what we find in the longest lived bats, they're much more enriched from the expression of anti-longevity genes. There's no difference in the pro-longevity genes, but genes that we know that if they're modified will decrease lifespan are expressed less as in longer lived bats than shorter lived bats. We also looked at autophagy. Again, we were taking wing punches from known-aged individuals of young, middle-aged, and older bats, of long-lived genera and of shorter-lived genera. We wanted to take these back in the lab, grow the entire transcriptome, and we wanted to test, as bats age, were they able to regulate their ability to remove protein and cellular damage? From our field studies, when you looked at what happens to these transcriptomes in the blood in known-aged bats, what we found that as bats age, they increase the amount of autophagy transcripts that they express. When we took this back into the lab, again, we grew the fibroblasts. We challenged them and we starved them to induce autophagy-like processes. And again, what we found was extraordinary. As bats age, they increase their ability to remove cellular damage. In mice, you don't see this. In us, we don't see this. So I know this was kind of a whirlwind, but the question is, how do bats defy the aging process? And what is their secret of extended health span? Well, they increase their DNA repair as they age. They increase their ability to remove their cellular damage. They maintain telomeres, at least the longest lived genera do. But also we found from looking at transcripts that they had a very balanced immunity. And potentially all of this is modulated by microRNAs and other epigenetic signatures. But to briefly touch on the concept of balanced immunity. Right now, we're in the middle of a horrendous global pandemic with over 180 million confirmed cases of COVID-19 and nearly 4 million deaths globally. Bats have been implicated in this because they are considered as a natural reservoir of coronaviruses and indeed contain the highest diversity of coronaviruses. But bats have been shown that they can live with these coronaviruses without getting sick. So therefore, studying bats also in an immunological context can give us insights in what we can evolve to allow us to live better with these viruses. But we were interested in this from an aging point of view. As we age, we become very, very inflamed. Have bats evolved a unique immune response that allows them somehow dampen age-induced inflammation, which also maybe could explain how they can live with all of these viruses? So what we were able to do was, working with our researchers in France on our fieldwork, we were able to capture a bat, a baby, that had lost her wing. She was euthanized by our vets. We were able to isolate stem cells, turn them into macrophages, and challenge them in the lab with these immune agonists to try and assess what is the immune response of bats to an infection. We were able to compare this with mice. And what we found with bats was that when they are mounting an immune response given an infection, they will very quickly mount a very aggressive antiviral response. But they then quickly match this with an equally aggressive anti-inflammatory response. And what you'll see is by the end of the simulation infection, they have more anti-inflammatory cytokines to inflammatory cytokines, which is very different than you would see in a mouse. Now, what this means, again, that means that bats have evolved mechanisms to allow them to modulate their immune response to have and limit inflammation. Now, the question is why? And again, I'm going to sum this all up. And again, this is to leave you thinking. Why have bats evolved all of these unique and different mechanisms that help them have a unique immune response that allow them live longer? Well, let's think about it. Flight is highly, highly metabolically costly, and it produces loads of endogenous stimulants. All the free radicals break up the mitochondria, cause damage to the DNA, cause lots and lots of issues, really overstimulate the immune response. So I'm going to argue that bats have had to evolve a unique adaptation of their innate immune response. They also have to evolve really excellent removal systems for cellular damage. They have to be able to repair their DNA. They have to be very quickly able to restore themselves and their cells to homeostasis. This allows them to deal with the metabolic consequences of flight and high metabolism. Essentially, they limit the idea of the scarb aging. What this has led to is unexpected longevity because all of their biomarkers of aging have to be maintained, and also a unique tolerance of pathogens, potentially as a side effect of flight. So I hope I've convinced you right now that for sure studying bats can allow us to understand how we can age more healthily. But not only this, that aging and infectious diseases are actually connected through looking at modifications of immune responses. And as we see these awful, horrible levels of death with COVID-19, that by studying bats will help us overcome both of these challenges. I would like to thank you all for listening and also thank the amazing group of researchers that I have to work with. Right now, I'm catching our long-lived bats in France, and hopefully I'll be able to talk to you. Take care.